Welcome back to part 2, 2.7. Let's go! So, here we have the nice image of DNA going to RNA, going to a protein or amino acid. And the special term for this first step, going from DNA to RNA, is transcription. So transcription, ooh, that's terrible. Let me write it better. In case some of you are relying on my handwriting to spell some of this stuff. So, transcription is DNA to RNA, and going from the RNA to the protein is translation. Translation. There we go. And real simple, it isn't always real simple, but DNA to RNA to protein. However, sometimes the DNA will just go off and do its own thing and have its own job in the cell, and it won't go to make any protein. But normally, DNA to RNA to protein. That's what the genetic code is based on. And to accomplish that, you need to know about this ribosome here, and we introduced this in the last video, ribosome. And here it is, cruising along your messenger RNA, and it looks for codons. See, it matched this up, AU and G and C. Remember how RNA has has a U instead of a T. So no T in RNA, only U's. So if you see a strand like this and you haven't even labeled or not labeled, whatever, you know it's RNA because there's a U right there. So what do you think is going to match up with this AAA right here? Think about it. What matches up with A? U, U, U. So that's called an anticodon that we came up in the last video with the tRNA right there. Swooping in, it's going to give its amino acid to this growing amino acid chain. Here's in a little more detail, showing how the position lines up. There's basically, there's three positions, essentially two, kind of three. You have the tRNA first come in, and then it shifts over. You briefly have two, and then the third, after it gives up its amino acid, it pops off. But up here is our polypeptide chain. It's growing. Polypeptide chain. Sorry, yeah, fan was making some noise. Polypeptide chain, and it's growing. And here is our tRNA. It's going to be entering, it's incoming. Incoming RNA, tRNA, and it's bringing that amino acid. That is an amino acid. It needs to match up, so eventually that will join there with a peptide bond, if you remember from last time. Peptide bond, yes indeed. And we also saw from that last image, the ribosome moves along. So it moves along that messenger RNA, translating, changing this message in codons into a message in amino acids. So we need to talk about some applications. How do we use this in real life? And this is how a lot of the coronavirus testing is going on right now. We're using RNA polymerase. Coronavirus is an RNA virus. RNA virus. Say COVID-19. It's easier to speak. It's faster, right? And it uses RNA polymerase PCR to figure out if you have it. So what happens here, this is how PCR works. But before I start that, it's real specific. It wants you to know about Taq DNA polymerase. And that's a special polymerase that comes from bacteria that lives in super hot geothermal pools. So if you're hanging out in a geothermal pool, you're going to need to cope with boiling water, basically. So scientists, when they develop this method, say, hey, we need a very hardy, very a polymerase that can withstand high temperature. So let's go looking at these bacterium that have evolved these polymerases over time to cope with the high temperature, because that's where you need PCR, because you need to separate the DNA. So you got to heat it to 95 degrees Celsius. That's almost boiling. Almost boiling. Got up to 100, it would be boiling. And that's called denaturation, this step. Because it separates the DNA, as you can see below, kind of broke it in half. Then you cool it down. And what that does, it allows primers, RNA primers, to bind to the template. It's called annealing. And then you heat it up a little bit. Your Taq polymerase is there. And that's called elongation. Because you're building, basically you're building new DNA right there. Boom. Look, from the single DNA, now you have 2 new strands. Pretty sweet, huh? Then you do it all over again, 30 times usually. And by the time you do that, I think you get something like a couple million DNA pairs. You see, this gets changed into these 2. So boom. In what? How many generations is that? 1, 2, 3 generations you get. 1 turns into 8. So this is how almost every forensic DNA technique is done. Any kind of DNA fingerprinting, cops go to an area, look for traces of any kind of human brain, blood, saliva, whatever it might be. They're going to be using polymerase, PCR, I mean, to increase the number of DNA samples, because it's really difficult to do analysis if you don't have a lot. And this is just an even more simplified diagram showing it. This is the original DNA, and this is after you denatured it here, and you'll see how the RNA primer is there. RNA primer is there, and that's the new strand. And you just heat it up again, and you get more and more. So that one initial DNA builds up into a huge DNA. Another really cool thing is, you've heard people that need to take insulin, they're type 1 diabetic. It used to be they had to get that insulin from pig pancreases and cows. So they had to go to slaughterhouses, get the pancreases, and then process. And very expensive, time consuming. You couldn't get that much. Plus, it's not even human DNA. It's from a cow or from a pig. Works pretty good, but a lot of people are allergic to it. So it doesn't work for a lot of diabetics. They found out a way in the 70s, I think it was the late 70s, to do it so you get actual human DNA. And the way you do is take a human cell and then any old bacteria, and any human cell will have the insulin gene in it. And it probably will be exactly the same. Like you and I probably have the same insulin gene. It'll be the same letters unless you're type 1 diabetic. I have a normal insulin gene, so I'm not. So ours will be essentially the same. It'll be the same exact letter coding. So we got the insulin gene. And then right here, this is called a plasmid. It's a piece of DNA that will go into a bacterium that can integrate itself into the bacterium host genome. So you mix up the insulin gene with this plasmid, you get what's called a recombinant plasmid, because it's recombined. And then you insert that back into a bacteria. Could have been this bacteria, could be another bacteria. And since you have genes from 2 different living things, that's now called transgenic. Transgenic, since you're going over 2 species, mixing their genes up. Then you want to culture it. It'd be a huge tank in reality, huge tank. But this gives you this kind of nice little thing. You got to culture it, to grow it. So just get it growing, and what it will do, it will start to make. This will start to produce human insulin. It'll read that information and then boom, start to pump out insulin. Insulin everywhere. And then you can extract it from here and then get it to people. So it's extracting insulin. Genetic code, written in codons. The groups of 3 we talked about before. You'll see here it gets changed subtly because RNA has U instead of T, but your body reads the U just like it would an A. And it is redundant. That means these 3 letters might code for one amino acid, each amino acid has a bunch of different letters that might be coded for, and I'll show you in a minute what that means. I can show you that next slide a little bit easier. Anticodon, of course, is what is on the tRNA, okay, and that helps to bind up the amino acid. And this is why the last thing worked. It's universal, which means all species use the same code. There are a couple of exceptions, I believe, in the domain archaea, where it is a little bit funky in terms of the way it translates, but more or less, remember, all rules have exceptions. And this one does too, but essentially all living things use this same code. And we're going to try this out. When I talk redundancy, you see this amino acid leucine, look, it has 6 different letter combinations that can code for it. Normally, you'll see the first 2 are the same. Because sometimes you get what's called wobble on this 3rd one. It won't quite, your body's trying to rush, and it's not even reading that last one. But it still normally works. It'll still give you the right one. Anyway, let's do this. Let's translate it. So if you had a codon ACC, what you're going to do is look over here. I found the A. So you're going to follow this row across, and then you're going to look for the C. So you're going to be looking in this set of boxes. And if you look at that last one, you can go, oh, matched up with that C. You can see that ACC, and it's threonine. All right. And then we'll try the CGU. Found the C. And then I'll look for the G. I go way over here. And then U. So CGU, arginine. Don't worry about knowing the names from the 3-letter thing. Not a big deal. Not a big deal. UAU. Got U. Go to A. And then find the U. That's tyrosine. Isn't this fun? ACG. You should stop the video and go ahead of me. That's what you should do so you can practice it better. ACG. ACG. It's also threonine. Like I told you, different codons, same amino acid. But you see, check this out. Got the same first letters. Make sure wobble happens. It's all good. GGA. GGA. That's glycine. UAA. So you'll see this is stopped. So this isn't even amino acid, but at the end of every gene, every mRNA strand, there's a stop. Tells your ribosome when to stop. When's the gene done? You gotta stop somewhere. You gotta know where to stop. Whew. So that's how your body knows, your cell, your ribosome knows when to stop. And then AUG, look for that. AUG. Oh, we'll see. See how it's highlighted? That's methionine. And that's special. It gets that highlight, that blue highlight, for a reason, because that starts everything. That's the start codon, start amino acid. Okay, very special. Every gene starts with MET. Okay, every protein chain starts with MET. All right, we did it.